What is cancer immunotherapy? Cancer immunotherapy is one of the most exciting and promising areas of cancer treatment today. Unlike traditional therapies like chemotherapy or radiation, which directly attack cancer cells, immunotherapy activates the patient's own immune system to recognize and destroy cancer. But how exactly does this work? Let's take a closer look. The immune system's job is to protect the body from harmful invaders, such as bacteria, viruses, and abnormal cells. It does this by identifying antigens or molecular flags that mark something as foreign. Cancer cells, however, arise from the body's own tissues. That means they can be harder for the immune system to detect because they often lack strong antigenic signals or actively suppress immune responses through the tumor microenvironment. One of the most powerful tools in the immunotherapy arsenal is the antibody. Role of antibodies in cancer? So, what are antibodies? Antibodies are Y-shaped proteins made by B cells, a type of white blood cell. They are produced in response to antigens, foreign molecules present on viruses, bacteria, and even cancer cells. In cancer immunotherapy, scientists engineer monoclonal antibodies that precisely recognize tumor-associated antigens, such as HER2 in breast cancer, R in colorectal cancer, or PD-L1 in lung cancer. Once an antibody binds to its target on a cancer cell, it can act in several powerful ways. 1. Blocking survival signals. For example, antibodies can block growth factor receptors, inhibiting the pathways that allow cancer cells to grow and divide. 2. Tagging for immune attack. Antibodies can recruit natural killer cells and cytotoxic T cells to destroy cancer cells via a process called antibody-dependent cellular cytotoxicity. 3. Boosting T cell activity. Some antibodies block immune checkpoints, such as PD-1, PD-L1, or CTLA-4, normally restrain T cells. This strategy, known as immune checkpoint blockade, allows T cells to stay active longer and attack tumors more effectively. Thanks to these mechanisms, antibody-based therapies have transformed cancer care. Examples include Keytruda, a PD-1 checkpoint inhibitor used in melanoma. Lung and bladder cancers. Herceptin targets HER2 in breast and gastric cancers. Rituxan binds CD20 on B-cell lymphomas and leukemias. These therapies are more targeted than traditional chemotherapy and often come with fewer systemic side effects. What's next? Scientists are developing bispecific antibodies, which can bind two targets at once, for example. One arm to a cancer cell and the other to a T cell, physically bringing them together. Another innovation is antibody-drug conjugates, which combine precise targeting with potent cancer cell killing by linking toxins or chemotherapies to antibodies, reducing damage to healthy cells. These next-generation antibodies are leading us toward highly specific and powerful cancer treatments. Antibodies have revolutionized how we treat cancer, and this is just the beginning. If you found this video helpful, please like, subscribe, and turn on notifications so you never miss new content about cutting-edge biomedical science.